This is Peter Brown from Bio 181. Today we're going to talk about genetics, how traits are passed from parent to offspring. A lot of students find this the most interesting part of the course. We're going to look at diploid organisms, organisms that have the same genetics as humans, but we're going to look at pea plants and Mendel's experiments, and what we say will be applicable to humans as well. Here's a nice picture of a monkey baby and the mother. So here's a little video about genetics. It talks about the units of inheritance called genes, and it's a 4-minute video, and it goes along with the Canvas quiz. Okay, this is a karyotype or a chromosome display. Like I said, we're only going to talk about diploid organisms, organisms like humans, and pea plants that have their chromosomes in pairs. You have chromosomes in pairs because one came from the mother and one came from the father. And what's nice about having chromosome in pairs, the genes are the units of heredity located along the chromosomes. The larger chromosomes have a few thousand genes. The smaller chromosomes have a few hundred genes. These genes are recipes for how to build proteins, which determine our traits. In any case, sometimes we get faulty genes. And if we get a faulty gene from one parent, usually the other parent has a good gene. So it's nice to have these combinations where a faulty gene can be covered up by a good gene. It's also interesting for sexual reproduction because you give one of only 2 of the chromosomes to your offspring and the others come from the other parent. So it's a way to shuffle up the genes in the next generation. So this kind of go through what I just told you in diploid organisms like peas and humans, and we'll do corn in lab. They all have pairs of chromosomes, and each parent forms sex cells, and they only give one of every pair to their offspring. So the sperm and egg have one of every kind of chromosome. They're called the haploid number, or one of each. And then the chromosomes are reformed when fertilization occurs, and then you're back to the diploid number. This fellow has a tragic story. His name was Gregory Mendel, and he lived in a monastery in Czechoslovakia, and he ran pea experiments in the back of the monastery. But he published them in an obscure German journal, and he didn't have a lot of money, so he didn't go to the scientific societies and advocate for his ideas. And so nobody really knew about his experiments until after he died, and somebody rediscovered his work, and they said, hey, this is the basis of heredity. So every genetics course in the world starts with the work of Mendel, a guy that was brilliant, but nobody even knew who he was when he was living. The good thing about working with peas is the male parts, the stamen, were large, as well as the female part. And so we could cut off the male parts on one flower and make sure that the pollen came from this plant, that this plant was the dad, and the eggs came from this plant, that this plant was the mom, so you could control which plants were the mom and dad. So here's a video that goes through in 3 minutes what we'll do in the next 15 slides. So you can watch it quickly or go through the slides and watch at the end. Okay, so he did these same experiments with 7 different traits. We'll go through the purple and white flowers, but you could go through and do it with all 7 traits. And first of all, he started with true breeding parents. So he let these plants self-pollinate until they bred true, which means that a purple flowered plant always gave rise to only purple flowered offspring. A white flowered plant only gave rise to white flowered offspring. And once he had true breeding parents for the trait he was examining, he crossed them together. And when he did, the parent generation produced the filial 1 generation, that's Latin for offspring, for sons and daughters. And when he crossed a purple flowered plant and a white flowered plant, he got all purple flowered offspring. That was pretty interesting in itself. But then he went a generation further, and he crossed the F1 generation with itself and with other F1 plants. And when he crossed these purple plants together, he got 3/4 purple flowered plants, but 1/4 white flowered plants. So he said, aha, in this generation that is produced by breeding a purple and white plant together, sometimes they're called the hybrid generation. In these hybrids, the white characteristic didn't disappear, it was just being covered up by purple, and we know that because it came out again here. So it's like maybe you have a characteristic that's not found in your parents, but it's found in one of your grandparents. The characteristic that covers up the other characteristic, we call that a dominant trait. The characteristic that gets covered up, we call that a recessive trait. In this case, purple is dominant and white flower color is recessive. So these were all the characteristics he looked at, and he found that same pattern in these characteristics, that one trait was dominant and one was recessive. And in the F2 generation, he had 3 of the dominant trait and 1 recessive trait. So when you get 2 different forms of the trait, we call that an allele, an alternate version of a gene. And if we think of a homologous pair of chromosomes, somewhere along that chromosome is a small piece of DNA that has a recipe for a gene. And here's a pair of chromosomes, and one parent gave the offspring a small b. So if we're following along, if it's a pea plant, that small b codes for white flowers, and a big B, the big B codes for purple flowers. Okay, so these alternate forms are called alleles. When we talk about whether the flower is purple or white, what we see, that's called the phenotype. And I remember that because the physical trait, what we observe, is the phenotype. But there are genes that represent that. And so on the previous slide, we saw that on the 2 chromosomes, there might be a big B or a small b on each gene. So the genotype is the 2 letters that represent the genes on each chromosome. So big B represents purple, little b represents white. So big B, little b, or big B would be a purple flower. Little b would be a white flower. Okay? So the letters, 2 letters represent the genotype, whereas the color is the phenotype. Okay, now let's look at his experiment showing the genotypes rather than the phenotypes. Okay, so since they're true breeding, the purple flower was big B. The white flower can only be little b. If it had a big B, it would be purple. And when you breed them together, each sperm or egg puts in a B in this flower. Each sperm or egg puts in a little b in this flower. And when the sperm and egg come together, you have to get a big B and a little b. So even though the F1 generation looks like the purple parent, the phenotype is the same, but the genotype is different. And when you breed 2 heterozygous plants together, you wind up getting 3 purple and 1 white. But again, even though we have 3 purple, they're the same phenotype, but you get 1 with 2 big B's and 2 with the big B, little b. And we'll show you how we figure that out in the next set of slides. Okay, so now we're going to do Punnett squares to see how we can figure all this out. And it was named after a chicken breeder in England named Reginald Punnett. And they're a way to represent the chromosomes and then the sperm and egg forming and then coming back together in the offspring. Okay, so for the parent cross, you're crossing big B by little b. Do a square, and on the outside, represent the sperm separating and the egg separating. It doesn't matter which one is the sperm and which one is the eggs. Okay, so the 2 big B's separate, the 2 little b's separate, and then in the middle, these represent the offspring. So big B across, little b down, big B across, little b down, big B across, little b down, big B across, little b down. So all the offspring are going to be purple colored. So the phenotypes are all purple, and all the offspring are big B, little b. So the genotypes are all big B, little b. Okay, when we do the F1 cross, we cross the first generation together, big B, little b by big B, little b. Do the same thing. Separate the big B, little b in the sperm, separate the big B, little b in the egg, and then bring them back together. Big B across, big B down. Big B across, little b down. Little b across, big B down. Little b across, little b down. Okay, so for the F1 cross, now you're crossing the offspring from the previous problem. 2 purple flowers, but now they're both big B, little b. So you separate the big B, little b in the sperm, you separate the big B, little b in the eggs, and then you bring them together. Big B across, big B down, big B across, little b down. Little b across, big B down, little b across, little b down. Then you count. Purple, 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 white. 3 purple to 1 white is the phenotypic ratio in the F2 generation. When you cross the F1 plants, you get the next generation, the F2. In terms of the genotypic ratio, we had 1 big B, big B. We can combine these two because they both have a big B, little b. It doesn't matter which you wind up writing first. So 2 big B, little b's and 1 little b, little b. That's the genotypic ratio. Okay, if they're both the same letter, but they're in the dominant form, then the plant genotypically is homozygous dominant. So instead of saying it's big B, we'll say it's homozygous dominant. If both the genes are in the recessive form, little b, it's homozygous recessive. And then if you have one of each, a capital letter and a little letter, then it's heterozygous. Hetero means both. When you write heterozygous, you don't use the words dominant or recessive because heterozygous is telling you you have one of each. So Mendel's law of dominance stated more formally, in a heterozygous individual, one trait is dominant and it conceals the presence of the second trait, recessive. Both traits are found genotypically, but only the dominant trait is seen in the phenotype. So a big B, little b plant is purple. You only see purple in the phenotype, but the genotype carries that white allele, which is covered up by the purple allele. All right, so here's one for you to do. Cross a purple pea flower that is heterozygous with the white pea flower. To get you started, what you're doing is big B, little b by little b, little b. And then after you've done it, give me the phenotypic ratio and the genotypic ratio. I'll have you do this on the homework. All right, if the whole world followed Mendel's rules, the world would be pretty boring. It turns out that if he saw a flower that was red and white, he said, okay, I'll look at that trait. But if he saw flowers that were red, white, and pink, he said, oh, this is too complicated. I don't want to do this one. It's too complicated. And most of the world actually works like this. This is more common than Mendelian dominance. If you get a true breeding red parent in snapdragons and a true breeding white parent in snapdragon flowers, and you cross them together, the offspring are all hybrids. They have a red allele and a white allele, but they're all pink. And that's called incomplete dominance. So to explain incomplete dominance, a gene is a recipe for a protein. And the phenotype depends on the interaction of proteins. So if you have Mendelian dominance, it means one protein is covering up the other protein. It's like spilling red paint on a white t-shirt, and if you spilled it all over, you might not even know it's a white t-shirt. It just looks all red. But if the proteins interact in a different way, it's like going to Home Depot and having them mix red and white paint together and making pink. And you can't really predict in advance if alleles are going to be dominant or incompletely dominant. You just have to do these crosses, look at the offspring, and then you say, well, based on the offspring, it shows complete dominance or incomplete dominance. So go ahead and do that cross. Big R, little r by big R, little r. In this case, if you have big R, little r, you have pink. Little r, you have white. And big R, you have red. Do the cross and then read the phenotypic ratio and genotypic ratio. And I'll have you do this on the homework. All right, recessive disorders. So there are some disorders in humans and plants as well, but humans is sort of an easier one to look at, that follow Mendelian inheritance. And so if you have 2 copies of a faulty gene, you have the disorder. If you have 1 good gene and 1 bad gene, the good gene covers up the bad gene. So that's why it's nice to be a diploid organism and have 2 copies of every gene. So here's a disorder that's unusual, although somewhere on TV or maybe in your own life, you've seen somebody who was an albino. Albinism is if you don't make any melanin. So people like myself, say, I have white skin. Yeah, I have white skin, but I still make melanin. It's just a different form of melanin than people who have darker skin. People who are albinos don't make melanin at all because of a defect. Okay? Generally, because these recessive alleles are rare, usually the parents are carriers. Carriers mean they're heterozygous. If you're heterozygous where the recessive allele is a disease, people call you a carrier. So her mother is definitely a carrier, and we'll assume the father's a carrier. So if 2 carriers mate, what are the chances that the offspring will have the disorder? All right, how about humans? It turns out humans aren't really very good to do genetic studies in. First of all, it takes about 20 years for a mother to have a baby, so the generation time is very long. Second of all, it would be unethical to say, okay, you mate with you, you mate with you, you mate with you, right? You can't do that. So you can't control genetics in humans, but one thing you can do is you can go back and follow the pattern of a trait within a family. And that's called a pedigree. Just like dogs have pedigrees, humans have pedigrees. So here's just a pedigree for a disorder for 2 generations. Here are the parents, here are the offspring. Square always represents males. So men are kind of nerds, they're kind of squares. And circles always represent females. They'll tell you which is the affected and which is the unaffected. In this case, they use blue for the affected and yellow for the unaffected. Okay, so here are the parents, 1 and 2, and here are the offspring. And go ahead and take a look at that and see if you can figure out what the genetics are for the father and the mother. You should be able to. If you can't, just read the next slide and it will explain it. Okay? Okay, hope you enjoyed that. We'll do a whole lab on genetics. So the lab will go right along with that, the genetics of corn. So if you didn't quite get it, we'll straighten everything out in lab.